during the chat, using the chat during Amar's presentation, using the chat and Q&A boxes that are on the bottom of your Zoom screen. We'll be collecting all of your questions and answering them, at least answering as many as we can, given how much time we'll have at the end, towards the end of the webinars, right after Amar is done with his presentation. We're going to attempt to stop at noon. I know we started a little late here, so if we run a little over noon, I think that will be fine for today's webinar. With that, let me get to introducing Dr. Amar Sahay. It's an absolute honor, Dr. Sahay, Amar, to have you here today. Amar is an associate professor at the Center for Regenerative Medicine at MassGen, Massachusetts General Hospital. He's also the James and Audrey Foster MGH Research Scholar, and he's also an associate member of the Broad Institute of MIT and Harvard. The mission of the Sahay Lab is to generate fundamental insights into the contributions of circuit and plasticity mechanisms in the dentate gyrus DGCA3 circuit, in hippocampal memory processing, and in the modulation of mood. His research has the potential to elucidate strategies to treat PTSD, Alzheimer's, social memory disorders, and age-related cognitive decline. So very translational in its context. Amar is recognized as a leader in the field of circuit neuroscience. He's been named as one of America's innovative new scientists by the National Institute of Mental Health and NIMH. We at Inscopix have also had the distinct honor to support his research and his lab's work through the Decode Grant program that Inscopix launched in 2014 in partnership with the Obama White House Brain Initiative. Today, Amar will discuss his group's work on how the hippocampus relays contextual information to subcortical circuits via the dorsolateral septum to calibrate defensive behaviors. Very, very interesting work that I'm sure all of you will be fascinated to hear about. So with that, Amar, the virtual stage here is yours.
Thanks, Kunal. Good afternoon. Good afternoon, everybody. Good morning. Good evening. Again, thanks for the invitation to speak as part of this exciting webinar series, especially during these very challenging times. I don't have any conflicts of interest to declare. Like many other academics, we use the Inscopix platform to record and analyze calcium transients and define populations of neurons. As an introduction, I think it suffices to say that context, or our environment, dictates our behavior, both at the level of physiology as well as action. Consider these two street scenes. The one on the left may evoke a sense of exploration, both spatial and social, may increase our walking cadence to explore. The one on the right, on the other hand, may make us more wary, reluctant to explore, may increase our skin conductance, heart rate. Needless to say that these behavioral adaptations are guided by our past experiences, registered as episodic memories, memories of what, when, and where. And these memories are continuously updated by changes in our environment or context, as well as goals. So for example, with the current pandemic, the street scene on the left, we may be no longer as keen to explore it. The hippocampus is thought to play a critical role in the formation of episodic memories, memories of what, when, and where. And it's thought to do so by integrating highly processed sensory information or context of our environment that arrives from higher order cortices to generate conjunctive representations that link objects and events with spatial and temporal contexts. And this information is then relayed to subcortical circuits to guide physiology and behavior. Circuit mechanisms in the hippocampus, such as pattern separation, ensure that similar memories or experiences, such as these two street scenes, the bottom left, are encoded as distinct representations, thereby decreasing interference between those two similar memories and preventing one from overriding the other. Pattern completion is another circuit mechanism that allows us to recall or retrieve a full representation based on only a subset of features in our context. So for example, this brick in the arch, through pattern completion, allows us to retrieve the entire representation of that street. Over the last 8.5 years, my lab has contributed to our understanding of circuit mechanisms in the hippocampus that support memory discrimination and memory generalization. Kate McAvoy, when she was a postdoctoral fellow in the lab, identified a role for adult-born neurons in encoding similar contexts in non-overlapping populations of neurons in the dentate gyrus. Tara Ram, when she was a graduate student in the lab, identified a physiological role for oxytocin receptors in the dentate gyrus CA3 circuit in the discrimination of social stimuli. Nanan Guha, who's currently a postdoctoral fellow in the lab and who's on her way to starting her own lab, recently identified a causal role for feed-forward inhibition in the dentate gyrus CA3 circuit in governing the extent to which memories generalize with the passage of time. And Antoine Benard, who's also a postdoctoral fellow in the lab, identified a transcription factor that mediates the effects of stress on fear generalization. And we'll hear more about Antoine's contributions in the seminar today. The linkage of contextual information with subcortical circuits to guide action, it's intuitive to think how inefficiencies or aberrations in this process may give rise to maladaptive, motivated, and defensive behaviors that characterize numerous psychiatric disorders. And we've spent a lot of time in the lab thinking about how computations about context that are made in the hippocampus are relayed to subcortical circuits to guide behavior. As many of you are aware, the hippocampus, as you move from the septal pole to the temporal pole, dorsal or ventral, has distinct patterns of efferent connectivity with brain regions. So the dorsal hippocampus, for example, has connectivity with the anterior thalamic nuclei, as well as robust connectivity with the lateral septum. The ventral or the temporal hippocampus has robust connectivity with the hypothalamus, the nucleus accumbens, the prefrontal cortex, and the lateral septum. When Antoine Benard joined my lab, he chose to be agnostic about which of these pathways may be preferentially recruited and asked the simple question, which pathways are recruited to link computations about context made in the hippocampus with subcortical circuits to guide contextual fear discrimination. And to accomplish this, he took two groups of mice, black 6 male mice. The first group was conditioned in a context A with mild foot shocks. He then challenged this group to discriminate between context A and a highly distinct neutral context, context B. He then took a second group of mice and challenged them to discriminate between context A, where they received the mild foot shocks, and a highly similar context, context A prime. As evidenced in these line plots, the first group exhibited high levels of discrimination between context A and this distinct neutral context. You can see high levels of freezing in the training context A and low levels of freezing in context B. The other group was unable to discriminate between these two highly similar contexts, the context A and the neutral similar context B. A prime, I'm sorry. Antoine then took these groups of mice, and after exposure to either context B or context A prime, he then perfused these animals and in a tour de force analysis mapped all the different brain regions by CFOS immunohistochemistry to identify regions that are highly sensitive to context discrimination and regions that are insensitive to context discrimination. He then took this data and generated a correlation matrix to identify brain regions that are highly coactive when animals perform high levels of discrimination. What we found, to our surprise, was that a dentate gyrus CA3 dorsolateral septum circuit was highly sensitive to the animal's context discrimination performance. This is in contrast to hippocampal CA1 that appeared to be largely insensitive to whether animals exhibited high levels or low levels of discrimination. As shown in the next slide, you can appreciate how high levels of activity in CA3 and in the dorsolateral septum are correlated with low levels of freezing behavior. This is not true, for example, with activity in CA3 and the amygdala. Now, this finding was made in, let's say, early 2013. We were very excited by this observation because it reminded us of pioneering contributions made by Larry Swanson using neuroanatomical tracing approaches that essentially characterized robust, massive projections from different subregions of the hippocampus, beautifully topographically organized within the dorsolateral septum. Since then, this is an example now of a single neuron reconstruction, a single CA3 neuron. And what you can appreciate is the terminal arborization of the single CA3 neuron within ipsilateral CA1 in red and contralateral CA1. So this is the Schaefer collaterals. And you can see the rostroventrally projecting axons that terminate in the dorsolateral septum. The lateral septum, in turn, has extensive connectivity with many different subcortical circuits. And this includes, so this is the dorsolateral septum right here. And this includes the BNST and the hypothalamus. Over the last 6 or 7 decades, classical lesioning studies and more recently, more refined approaches in neuroscience, circuit interrogation approaches, has implicated the lateral septum in the regulation of emotion, in aggression, and in reward-seeking behavior. In 2011, we were inspired by what we think was a landmark study that came out of Gary Aston Jones's lab that identified a CA3, hippocampal CA3, dorsolateral septum VTA circuit as important for contextual reinstatement of cocaine-seeking behavior. More recently, in vivo recordings in the hippocampus and in the lateral septum in awake behaving rodents has identified theta rhythmic firing of LS neurons and has suggested roles for the LS neurons, lateral septal neurons, in both conveying spatial information as well as the regulation of speed. So what Antoine sought to address first and foremost was whether or not CA3 projections, as you move along the septotemporal axis of the hippocampus, subscribe to any pattern of organization within the dorsolateral septum. And what Antoine found is that dorsal CA3 projections terminate exclusively in this medial region of the dorsolateral septum as shown over here. In contrast, ventral CA3 neurons terminate in this lateral region of the DLS, reminiscent of what Larry Swanson had shown and characterized in the rat. Antoine then asked, what is the functional contribution of these projections from CA3, dorsal and ventral CA3, to the lateral septum, to contextual fear discrimination learning? And to address this, he expressed heterodopsin in either dorsal CA3, and these are the terminations, the axon terminals in the medial region of the DLS, and in a separate group of mice, expressed heterodopsin in ventral CA3 neurons, and you can see their terminations over here in the lateral region of the DLS. Antoine then ran these animals through a contextual fear discrimination learning assay as shown in the bottom of the screen. Animals were conditioned to a context A, where they received mild foot shocks, and then challenged to discriminate between context A and a highly similar context that's neutral, context B. After several blocks of training, animals reach an asymptotic level of behavioral performance, of discrimination between context A and context B. Antoine then silenced dorsal CA3 projections in the DLS when the animals are placed back in context A or context B, and he did the same with the other cohort of mice, silencing the projections of ventral CA3 neurons when the animals are placed back in context A and context B. What Antoine found is that when you silence dorsal CA3 projections in the dorsolateral septum, you don't affect freezing levels in the fear-conditioned context A. Instead, we observed a reduction in freezing levels in the similar context B. And this in turn impaired context discrimination as captured in the discrimination ratio. In contrast, when Antoine silenced ventral CA3 projections to the DLS, this is in a separate group of mice, again using the same contextual fear discrimination learning paradigm. So once animals have reached a stable level of discrimination performance across several blocks, to then silence ventral CA3 projections when the animals are placed in context A or context B, Antoine found that this resulted in elevation in freezing levels in both context A as well as in context B. These observations, along with other interrogations as part of this study, allowed us to generate a course framework for thinking about how dorsal CA3 and ventral CA3 projections to the DLS calibrate contextual fear discrimination. Based on other aspects of the study, our current thinking based on this model is that when animals are still learning to differentiate or distinguish between the fear-conditioned context A and the similar context B, then CA3 projections in dorsal CA3 to the DLS play a role in promoting freezing behavior. And this is because the animals have not generated a conjunctive representation of context A as yet. Once the animals are able to differentiate context A and context B, and this is after generating a representation of context A through the dorsal CA3-CA1 prefrontal cortex pathway that mediates memory consolidation, then the dorsal CA3 to DLS projection suppresses freezing in the similar neutral context, because the animals now can differentiate the fearful conditioned context A from the similar neutral context B. So CA3 projections to dorsal CA1 and DLS have distinct roles, we think, in consolidation of context A and regulation of freezing levels in the neutral context B to mediate discrimination. Ventral CA3 projections to the DLS, on the other hand, appear to be insensitive to contextual information and sort of function like a gain control mechanism to change or modulate levels of freezing in both context A as well as in the similar neutral context B. During the course of these studies, when Antoine silenced dorsal CA3 projections in the DLS or ventral CA3 projections in the DLS, we observed widespread changes in downstream subcortical circuits as assessed by CFOS immunohistochemistry. These observations motivated us to think about what are the identities of the cellular relays within the DLS that link computations about context in the dorsal hippocampus and in the ventral hippocampus with all of these different subcortical circuits, such as the PAG, the BNST, the lateral hypothalamus, and the supramammary nuclei, for example. As many of you may be aware, the lateral septum is made up of primarily GABAergic neurons that are enriched or express exclusively distinct neuropeptides. In today's talk, I'm going to focus on two populations of lateral septal neurons, the somatostatin class and the parvalbumin class of inhibitory neurons. The parvalbumin class of inhibitory neurons are highly localized within the DLS and in that region where dorsal CA3 projections terminate. It's very, very sparse, the distribution, and these PV cells in the DLS project both locally within the DLS, as well as have these long-range projections to the BNST, the lateral hypothalamus, these beautiful termination zones in the supramammillary nuclei. Using pseudotype rabies tracing, Antoine identified putative presynaptic partners of these PV cells in the DLS in CA3 and to a great extent in CA1. In collaboration with Samara Miller, a postdoctoral fellow in the lab, Samara found that following optogenetic stimulation of dorsal CA3 terminals in the DLS, so this is ex vivo slice preparation, she could identify monosynaptic evoked excitatory responses in genetically labeled PV cells, suggesting that these PV cells in the DLS receive functional synaptic inputs from dorsal CA3 neurons. What is the contribution of these PV cells to contextual fear discrimination learning? To address that, Antoine took a separate cohort of mice where he expressed halorhodopsin in the PV cells of the dorsolateral septum. He then challenged these animals to discriminate, as I've shown you previously, between a fearful context, context A, the animals are conditioned by virtue of receiving mild foot shocks in that context A, and a highly similar context B. And again, once behavioral performance becomes asymptotic or stable and animals exhibit discrimination, Antoine then silenced the PV cells in the dorsolateral septum when the animals were placed back in context A or in the similar context B. What Antoine found was that this did not affect freezing levels in context A, but only decreased freezing levels in the similar context B, thereby impairing context discrimination and is captured in the discrimination ratio shown over here. Now you might recall that this is exactly what we saw at the level of behavior when we silenced dorsal CA3 projections to the DLS. If I just toggle back and forth between these two slides, you can appreciate the similarity in the behavioral outcomes of either manipulating or silencing PV cells in the DLS or silencing dorsal CA3 projections in the DLS. We next turned our attention to the more abundant somatostatin class of inhibitory neurons in the DLS. Somatostatin cells comprise the largest population of GABAergic neurons in the dorsolateral septum. And like PV cells, they have projections that are local as well as distal to different subcortical targets, including the BNST, the lateral hypothalamus, and the supramammillary nuclei. We first asked whether these somatostatin cells receive synaptic inputs from CA3 neurons. And again, Antoine performed pseudotype rabies retrograde synaptic tracing to identify presynaptic partners, these genetically labeled SST cells in the DLS, presynaptic partners in CA3, CA1, as well as subcortical inputs and prefrontal inputs. In collaboration with Ian Davison and Yuan Gao, we established that these SST cells in the DLS receive functional excitatory inputs from hippocampal CA3 neurons. We then asked, what conditions recruit this population of SST cells in the dorsolateral septum? And to do this, we expressed GCAMP in somatostatin cells, and using the miniaturized endoscopes and the platform available from Inscopix, we then proceeded to record calcium transients in these SST cells in awake behaving mice. And these experiments were led by Antoine Bernard and a very talented Michael Kim, a college graduate who is now doing his PhD at Harvard University, and Hannah Torkowski, a postdoctoral fellow in my lab. What Antoine, Michael, and Hannah did was run the animals through a behavioral paradigm shown over here. The animals first went through behavioral assays, or will run through behavioral assays that probe anxiety-like behavior, like the open field and the elevated plus maze. But let us focus our attention on contextual fear learning. The animals are then conditioned to a context A where they received mild foot shocks. They were then 24 hours later placed back in that fearful training context, context A, or a distinct neutral context, context B. A few days later, Antoine then returned these animals to context A, where they received mild foot shocks. And the reason we did this was to determine if the allocation of somatostatin cells during contextual fear conditioning was stochastic, or whether there might be a specific population of neurons that are preferentially recruited in contextual fear conditioning. And this will become apparent in the next few slides. So this is now the behavior of the animals. Prior to conditioning, the animals express high levels of mobility, very low levels of freezing, if any, in context A. Following conditioning, 24 hours later, when the animals are placed back in the context A, you can appreciate that now the mobility of the animal is highly regulated. Behavioral state transitions are very marked. That's mobility and immobility. The animals are mostly immobile and then transition into a mobile state and then return to an immobile state. In contrast, in the distinct context B, the animals are very mobile with very, very few bouts of immobility. So what did Antoine and colleagues find with observation, a recording of calcium transients in these somatostatin cells during contextual fear learning and recall? What Antoine and colleagues found, to our surprise, is that there appears to be a stable population of somatostatin cells that are responsive to foot shock during contextual fear conditioning 1 and the second contextual fear conditioning episode, CFC2. There appear to be somatostatin cells that are insensitive or not recruited during contextual fear conditioning, as they are not recruited in either CFC1 or CFC2. And this population of cells, what we are referring to as the somatostatin foot shock sensitive cells, the FSH cells that respond to foot shocks, the overlap between CFC1 and CFC2, as ascertained by both PCA, ICA, and CNMFE, is distinct from that of a null distribution. And this is also true for the non-foot shock sensitive cells. This suggests that not all somatostatin cells may be the same or may be homogeneous. Rather, a potentially stable, hardwired population is preferentially recruited during contextual fear conditioning. What is the behavior of these cells now when we place these animals back in the fearful context A 24 hours later? When Antoine did that, what we found is that the foot shock sensitive cells, the somatostatin foot shock sensitive cells that we had identified, their activity tracks the mobility-immobility transitions, behavioral state transitions, when animals were placed in this context where animals had to regulate their freezing levels and mobility levels to a much greater extent than they did in context B, for example, where the animals are very mobile. You can appreciate how the activity of the SST foot shock cells is high when the animals are moving, so not freezing. And when the animals begin to freeze, the activity of these cells ramps down. When the animals become mobile again, the activity of these cells ramps up. The non-foot shock sensitive cells did not exhibit this activity tracking of behavioral state transitions. Furthermore, this property of somatostatin foot shock cells was seen in the conditioned context A, where the animals had to tightly regulate their mobility, rather than in context B or in the open field, where the animals are free to move around and were doing so. This suggests that somatostatin foot shock cells may be a stable population, potentially hardwired, to participate in contextual fear conditioning and recall. This finding was reminiscent of pioneering single unit recordings in the lateral septum performed in the early 1990s, where a population of cells in the LS was identified as highly sensitive to the presentation of aversive conditioned stimuli. Their activity went down when the aversive conditioned stimulus was presented, and their activity ramped up when this aversive CS was removed. So what is the contribution of these somatostatin foot shock cells to contextual fear learning? To address that, Antoine then expressed haloviropsin in SST cells in the dorsolateral septum. And after he conditioned animals to context A, he then, as I've shown you before, he then silenced these SST cells and the animals are placed back in context A or a similar context, context B. And what he found was he found widespread changes in downstream subcortical circuits, including the LH, the SUM, the BNST, the PAG, periaqueductal gray. And behaviorally, he found that there was an elevation in freezing levels in both context A as well as the similar neutral context, context B. Now, you might recall that this is precisely the behavioral outcome when we manipulated, when we silenced ventral CA3 projections to the DLS, which is what I'm showing you now. So if I toggle back and forth again, you can appreciate that silencing SST neurons in the DLS or ventral CA3 projections in the DLS elevates freezing levels in both context A and context B. Based on these observations, we can now update our framework for how we think about how dorsal CA3 and ventral CA3 projections in the DLS calibrate contextual fear discrimination. We think that parvalbumin cells in the DLS and somatostatin cells are putative relays for dorsal CA3 projections to link contextual information to calibrate freezing responses in similar contexts, and ventral CA3 projections to calibrate freezing levels insensitive to contextual information, but sort of like a gain control mechanism. The functional imaging studies that I've shown you have alluded to the possibility that not all somatostatin cells are the same, and that subpopulations, such as the foot shock population, sensitive population that we've identified, an open arm sensitive population that we've identified, may be molecularly prescribed. And that distinct subpopulations of somatostatin cells in the DLS, by virtue of their genetic prescription, may have distinct topographic organization of both inputs, either just from the dorsal hippocampus or the ventral hippocampus or both, and to distinct subsets of subcortical targets. As a first step to understand the molecular basis of this hypothesized SST heterogeneity that we've surmised from our imaging studies, we entered into collaboration with Evan McCosco's lab at the Broad Institute of Harvard and MIT. And we drop-seeked 27,000 cells from the dorsolateral septum. Following transcriptomics of single cells, we identified 7 putative subpopulations of somatostatin cells enriched in distinct neuropeptides, proenkephalin, neurotensin, with distinct topographic organization of their cell bodies within the rostrocaudal and dorsoventral axes of the dorsolateral septum. So now we've assembled an exciting team of Samara Miller, Travis Good, Aing Chung, and Antoine Bernard. And Samara is now investigating whether these putative somatostatin subtypes using neuropeptide, CRE, and SST flippase combination lines exhibit diversity of electrophysiological properties as has been beautifully documented for SST cells in the neocortex. And she's mapping the local connectivity within the dorsolateral septum. Using many of the approaches that I've shown you, calcium imaging and in vivo physiology now, we hope to generate a blueprint for how computations performed in the dorsal hippocampus or ventral hippocampus are relayed through distinct subpopulations of somatostatin cells, some of which might act as parallel channels segregating the flow of information from the hippocampus to subcortical targets, and some of them multiplexing information from dorsal and ventral hippocampus or different subregions within the dorsal hippocampus. Two distinct or overlapping subcortical circuits, such as the BNST, the LH, the nucleus accumbens, periaqueductal gray. And to calibrate not just defensive behavior like I've shown you, but motivated behaviors, reward, food, feeding, and we plan to move away from freezing, as we've used primarily as a readout, and employ a statistical deconstruction of behavior through some of the modules that are now widely available in the academic community. Last, I'd like to share with you the conclusions and the working model. We've identified a CA3 DLS circuit that's highly sensitive to context discrimination, a role for parvalbumin cells and somatostatin cells in the dorsolateral septum as putative relays of dorsal CA3 and ventral CA3 projections to the DLS to calibrate contextual fear discrimination in distinct ways. We posit that these SST cells are arbiters of mobility that calibrate context-appropriate responses underlying both defensive and motivated behaviors, and that distinct subpopulations of somatostatin cells characterized by distinct patterns of input-output connectivity, physiology, and neuropeptide expression play a role in this calibration of defensive and motivated behaviors by our context or our environment. Last but not the least, I want to thank the folks in my lab. This project was spearheaded by a very talented postdoc, Antoine Bernard, who's now interviewing in INSERM and CNRS, and I think any institution in France where he's looking will be very lucky to have him as an asset, as a colleague, and as a scientist. It's really been exciting sharing this journey with him in my lab. I've mentioned the names of the other folks who've contributed to this project, our collaborators, our funding agencies, private funding, as well as the NIH. This project was really kickstarted by a BRAINS R01 that I received 5.5 years ago. And I'd like to thank other funding agencies and MGH Psychiatry for their support as well. I would be remiss if I did not, during these really challenging times, acknowledge the contributions of my friends and colleagues at Mass General, who are first responders, who are in the front lines, and all the other first responders in hospitals, in grocery stores, bus drivers, because of whom, we can really resume or try to resume our science with a sense of normalcy. So with that, I'll end this presentation, and I'll be happy to take questions. I'll thank you for your time and for listening.
Thank you, Dr. Sahai. Hi, everyone. My name is Sushmita. I work in the marketing team at Inscopix, and I'm jumping in to moderate what I'm hoping will be a lively Q&A discussion. At this time, I want to encourage everyone to submit any questions they might have on the talk using the Q&A or the chat windows at the bottom of the screen. We welcome all and any questions. Okay, so Dr. Sahai, thank you so much again for this beautiful story. Contextual fear discrimination is certainly something we can all, as human beings, relate to, I think, on a daily basis almost, especially during these times. And the sophisticated subcircuitry that you shared was really helpful in understanding how our brain processes these experiences. So we've started to get some questions, so we can jump in. We have Saktival Srinivasan, I hope I'm saying that correctly, asking if you ever recorded simultaneously silenced DCA3 and also image in the DLS using the invoke.
Yeah, it's a great question. So we plan to do that with these defined subtypes that we've identified. It's a great question.
Mm-hmm. And then I think related question from the same person. Did you ever stimulate the SST and image the CA3 by invoke?
We have not done that. I mean, we don't see reciprocal connectivity between SST cells and CA3 neurons. So we have not done that experiment.
Okay, thank you. Moving on, Jesse Cushman has a comment on the sex differences. Were the studies done only in males is the question.
Yeah, it's a great question. So insofar, yes, the studies that I've shown you, but as you are probably aware, the LS is interesting for many reasons, including that it's sexually dimorphic. And moving forward, again, now that we have a much granular map of these somatostatin cells, we are going to be paying close attention to whether the characteristics of these distinct SST subpopulations subscribe to male or female boundaries to get at your question. Yeah.
That makes sense. Okay, so Pablo Fuentealba, hope I'm saying that correctly, is asking what would happen if you silence or activate SST positive cells in the DLS?
If we silence or activate SST positive cells in the DLS?
Correct.
Yeah, so that's exactly what we did. We activated SST cells and we silenced SST cells. What I showed you was the silencing of SST cells during contextual field learning. But we've also activated these cells and silenced these cells in behavioral assays that probe anxiety-like behavior, and contextual field learning. So for example, when you silence the SST cells in the open field, we don't see any effects. And we actually don't see any effects when you activate the SST cells in the open field either. So all of these data actually are published in that Nature Neuroscience paper. If I understand your question correctly.
That question was prefaced with maybe I missed it. So actually that might have been.
Yeah. What is the question again then?
Oh, I think you answered the question.
Okay.
I think the questioner might have missed some section of the talk. Okay. Yeah.
Well, once it's recorded, you can revisit it.
Yes, yeah, for sure. Okay, so we have a comment saying what a wonderful talk, agreed, and a small question. The SST immunostaining in the DLS has a dorsal ventral gradient. So does this gradient map on to afferent patterns or functional activity differences?
Yeah, so it's a great question. I mean, you're right. It is more enriched ventrally than dorsally. But whether or not within the SST population, looking at SST, there are differences in hippocampal outputs as you move from the septal to the temporal pole. We don't know as yet. But I think, again, now with this granular map that we have, we know that now, we're focusing all our attention now on these SST subpopulations, right? And so those have their own distribution. So the neurotensin has a distribution, the many labs working on neurotensin, for example, but the intersection of SST and neurotensin, SST and proenkephalin, for example, whether these subpopulations, which we think are different from each other, and maybe so physiologically as well. This is something that Samara is actively exploring, whether they have differences in dorsal and ventral inputs. we are actively exploring that question. So rather than looking at SST, we're looking at the subpopulations of SST that we've identified to ask precisely what you've asked me, whether they have differences in dorsal and ventral, but also within the hippocampus. Within the dorsal hippocampus, for example, is there a bias towards CA1 versus CA3 versus subiculum?
That's really interesting. Yeah, we hope we get a peek at that soon. Okay, Kimberly Long asks, does the DLS act as a relay only for the hippocampus, or does it integrate input, either neural or neuroendocrine, from multiple regions?
It's a great question. So the DLS, these neurons receive inputs from the prefrontal cortex. There's some beautiful work by Natalia Korotkova, who's at the Max Planck in Cologne, showing the role for prefrontal cortical inputs in reward-seeking behavior and in calibration of approach towards reward. These SST cells, like I've shown you, receive inputs from the lateral hypothalamus, very intriguing. And we're seeing that with some of the subpopulations as well, suggesting that they integrate. So yes, I did use the word relay, but we do think that these DLS cells, SST and PV, integrate information from hippocampal, prefrontal, and subcortical. So there's reciprocal connectivity with subcortical circuits as well. And so I use the word relay, but we don't think they're just relays. They might be integrating information. So for example, within the hippocampus from CA3 and CA1 simultaneously, as has been suggested by Buzaki's lab, but now we can access these defined populations and ask that question. And to your point, the molecular access now gives us the permission to ascertain to what extent are neuroendocrine inputs integrated and for what.
That is, yes, that is. Thank you. That is really cool. The hippocampus definitely does many, many things as we continue to learn every day. Okay, so another question is, if you think whether the SST foot shock activated cells are analogous to engram cells identified in the dorsal CA1 or the MPFC during similar fear conditioning behaviors?
Yeah, so again, a great question. So we actually have a perspective piece that's under revision now, where we're talking about how engrams, by virtue of being distributed, may enable the hippocampus to recruit subcortical circuits to then calibrate different behaviors. So I don't think the LS cells per se are engram cells, but they may be participant to a much larger distribution that functions as an engram to support memory indexing functions.
Okay. Okay, so this seems related to some questions that you're already answering. So this is from Rebecca Sent. She asks, are you also considering examining other subcortical inputs that may target both the DLS and the DHP, such as the raphe nuclei?
Yeah, so we don't see in our mapping studies insofar we don't see inputs from the raphe.
Okay.
Yeah.
Okay, that answers it. Yeah. Let's see. So Nicola,
there are raphe inputs, as I know Rebecca is working on this onto populations of cells in the DLS. We just haven't seen that as yet.
Okay.
Yeah, for the cells we're looking at, the subpopulation.
Yeah. Right. Okay. So I was looking at another question from Nicola. First, thanks for the great talk. Are you doing any work to understand which SST receptors are mediating these effects?
Not insofar, but it's these are all great questions. Yeah.
Yeah. Okay, just gonna check on time. We're at 11:53, so we could take maybe 2 or 3 more questions. Let's see. We have a question that asks, Where do striatum and corticostriatal circuits fit in the proposed model involving the hippocampus and the DLS?
Well, I mean, it could be perceived as a parallel competitive circuit that's used once tasks become second nature and more procedural. But I mean, that's the way we're thinking about it now. I really haven't spent much more time thinking about corticostriatal circuits and in terms of how they are acting in parallel or sequentially to hippocampal dependent recruitment of downstream circuits.
Okay. All right. So
I mean, it would be interesting to see if prefrontal inputs to the LS, if they're collaterals to the striatum. I think that would be really exciting.
Yeah.
Lots of questions, and that would get at what you're trying to ask. We don't know the answer.
Certainly. Yeah. Okay, so we have a question from Romeo Go. He says, great talk. May I know why you chose to examine albumin positive neurons instead of other more highly prominent neuron types in the lateral septum?
Well, I mean, truth be told, I mean, when we saw when Antoine first noticed that these parvalbumin cells were in the DLS, it was at first surprising because there's a tremendous body of work on parvalbumin cells in the medial septum, and the parvalbumin population of DLS has been largely ignored. And to my knowledge, there's no function that's been ascribed to this really sparse population of PV cells in the DLS. So when Antoine found that, we were most intrigued by what these PV cells are doing in the dorsolateral septum, and also their localization instantly suggested to us that they may be preferentially relaying dorsal hippocampal outputs based on Antoine's anterograde projection mapping analysis in the mice, in the mouse. So those are some of the reasons why we pursued this sparse population of PV cells in the dorsolateral septum.
Yeah, that's a great rationale. Okay, so Alejandro Schinder, I hope I'm saying that right. Okay, yeah, he says, hello, spectacular talk, Amar. And the question is, what is the output network being controlled by those somatostatin cells sensitive to foot shock? They seem very powerful in controlling freezing because silencing them induces freezing.
Yeah, so which specific targets of these SST cells are mediating the freezing or mediating, calibrating freezing levels in the context, in fair condition context or the neutral context? We do not know as yet. But again, now that we've mapped the subpopulations, we're focusing on those to look at their contributions towards these readouts such as freezing. But like I said, we also want to go well beyond freezing and look at the micro modules that underlie defensive behavior and motivated behaviors.
Okay.
I mean, suspects like the periaqueductal gray, for example, that are likely to be substrates. So the SST projections on the DLS to the PAG that may be responsible for modulating freezing levels. But we have not done that experiment as yet. We will in these subpopulations of SST cells.
Yeah, okay. Great. So this is from Estefania Azevedo. Asks, have you looked if foot shock responsive SST cells also are tuned to other defensive behaviors, such as looming stimulus or opposite valence stimuli, such as rewarding?
Yeah, that's a great question. We have not done that experiment. And again, we're not looking at SST cells in the way that we have with calcium imaging right now, where we're imaging SST cells. We're now segueing into subpopulations of SST cells because it looks like these subpopulations may have distinct input and output connectivity, and so we're looking at those neurons and their responses to appetitive cues, rewarding cues, and threatening cues, or different modalities. Great question, though. Because
Yeah,
yeah,
it's really interesting. Yeah. Sorry, go ahead if you wanted to
no,
elaborate.
I think I answered the question. Yeah,
Okay.
great question.
Okay, so we're getting towards the end of our time slot here, and so I want to end with a big picture type question that's come in. The question is if you can comment on the implications that your work might have on our understanding of neuropsychiatric disease, such as anxiety disorders, or any others that you might be thinking about.
Yeah, I mean, I think we have the work, really establishes a foundation for thinking about how aberrations in our processing of context results in maladaptive behavioral responses. So, context-dependent relapse, whether it be drug reward seeking, whether it be PTSD, where you have fear generalization based on aberrant processing of contextual cues. So whether it be defensive behaviors or motivated behaviors, it's important to realize or think about how much there is contextual gating of these different behaviors. The way we interpret and process our context directly affects the way we respond behaviorally. And so if the pathways that link the hippocampus with any of these subcortical circuits that I've talked about are disrupted, then immediate prediction would be that this would result in inefficient linking of that context that's always changing around us with our experiences with the behavioral repertoire that we use to navigate our world. And so, that's why we don't like to say we focus on PTSD or only on autism, or if these circuits that we are focusing on and I've talked about, they are substrates for both defensive and motivated behaviors. And as such, we would predict that alterations in this linkage of context with these subcortical circuits, the BNST, the lateral hypothalamus, the supramammary nuclei, the PAG, when disrupted, would give you the excessive generalization, excessive expression of fear, or the miscalibration of behavior when you see something that's threatening, or when you see something that's not threatening, or the converse, when you perceive a contextual cue and that recruits a reward circuit to then produce relapse. All of this requires contextual gating. And so we think this work and this series of this roadmap that we're building now, all of these neuropeptides expressed in these different subpopulations, we don't know what they're doing. But our prediction is that it will have direct implications for our understanding of the origins of maladaptive, defensive, and motivated behaviors that characterize many different psychiatric disorders, from PTSD on one hand to relapse that's seen with reward-seeking behavior like drugs of abuse on the other hand, as well as our calibration of social interactions. So, I like to be agnostic in terms of, whether it's psychiatric disorder A versus B, but I think it's because an understanding at this level is likely to broadly impact how we think about the extent to which our world, our context, calibrates these physiological parameters, skin conductance, heart rate, as well as behavioral output. Great question.
Yeah, certainly a great way to close the talk by thinking about the translational applications of your beautiful work. So at this time, thank you so much, Dr. Sahai, for answering all the questions. I'd like to invite Kamal to offer some closing remarks.
Thank you so much, Amar, for a really, really fascinating talk. And it was a very nice note to also end thinking about, what are the translational ramifications of some of the research you had presented. And I totally agree. I think the beauty of circuit neuroscience is that in many ways it's agnostic to the specific disease that one is trying to tackle. So terrific way to wrap. Just for the benefit of the audience, as a quick reminder, we're of course going to continue this bi-weekly insight series. We are continuing to present a spectacular lineup of world-class neuroscientists like Amar over the next few weeks in June and July. The next few speakers, beginning with Ilana Witten of Princeton University on June 15th, followed by Michael Bruckus of the University of Washington on June 29th, and Zoe Donaldson focusing on the Prairie Voles from Colorado on July 13th. So we're continuing to do our best to cover a wide spectrum of topics from addiction to social behavior and present a diversity of speakers and species through this series. We'll be emailing you the signups for these seminars very soon, so please look out for them. We'll also be posting the schedule and topics for the seminar series as it continues to progress on all of our social media channels. If you find these webinars helpful, which I hope you do, please do help us spread the word through the global neuroscience community. We certainly, and our speakers would certainly appreciate all of you trying your best to help us reach a broader audience. Thank you again, Amar, for an exciting and really enlightening talk. And thank you to all of you for participating in today's talk and in this series. Take care, everyone. Stay safe.
Thank you, everybody. Stay safe. Stay healthy.